I'm a postdoc in the Whitehead lab at Harvard, and I'd like to thank the HFSP for the funding of this project, which is really exciting. And I'm going to be talking about the limb regeneration process in the axolotl, and how this might be regulated in part by how angiogenesis or blood vessel formation is regulated. What you can see up here right now is a 3D rendering of an axolotl limb, and it's highlighting the blood vessels in this in the limb. So, limb loss is a huge problem worldwide, and in the US alone, there are around 2.1 million people living with amputations, with 185,000 amputated every year, which equates to 507 a day on average. And this is a bigger problem throughout the world, of course. And not only do people with limb loss have to live life without a limb, they also are at increased risk of other diseases, including cardiovascular disease. And this seems to be irrelevant of whether the mobility of the person is affected or not. And actually, the life expectancy of someone who receives an amputation is only 5 years from amputation. So that's why we use the axolotl to study this. Since humans are not capable of regenerating a limb after amputation, we have a scar, and that's all we get. But salamanders have limbs like humans. They have digits, they have the same sort of bone structure, and they're capable of regenerating their limb throughout life. And here we can see this in action right now. So this is an axolotl, which you'll see in a moment. It has an amputation, and you'll see the blastema form before the limb forms. So right now we can see the blastema forming right here. This is a specialized structure which is required for limb regeneration, and then eventually it gives rise to all of the digits of the limb. And I'll let this run through twice just to point through things again. So now you can see that the limb is completely analogous to the other limb, and it's indistinguishable from an unamputated limb. So this is the blastema, which I'll talk about a lot. This is a specialized mass of undifferentiated cells, which can give rise to all the tissues of the limb, as you can see here. So, what is a blastema? It's an undifferentiated mass of cells, which have stem cell-like properties. They're mesenchymal and migratory. They're highly proliferative. But how do they form is the question, and what is this process? What is the process regulated by? Is what I'm interested in. And how do axolotls respond to sort of a missing tissue response? And especially when you compare this to organisms which scar versus those which regenerate. So mammals like ourselves and mice tend not to be as regenerative and scar more than other animals such as salamanders and zebrafish. So what's the difference here? So I think that angiogenesis might be part of the process. So in this example here, following an injury in a non-scarring organism, there'll be controlled angiogenesis, allowing the wound to heal before angiogenesis occurs, which results in no fibrosis. But in an animal that scars, there's a burst of angiogenesis which quickly allows platelets and fibroblasts to come in and patch up the injury, but this results in fibrosis. And we know that in axolotls, from the postdoctoral work of my supervisor, that anti-angiogenic signals are expressed in the wound epidermis during regeneration to prevent excessive angiogenesis. So, this is why axolotls probably don't scar. But can they scar? And the answer is kind of, actually. So if you take an axolotl and you repeatedly amputate it, eventually it stops regenerating. And you can see here it just has a stump. Over the course of several amputations, the chances of this happening increases. So if you take a look at the tissue sections as well, in a control animal on the left, you see that there's an organized skin layers with collagen in blue here. And then if you look at one that's received multiple amputations, you see that this organization is lost, and you see excessive collagen deposition, which is kind of scar-like. As we've seen earlier today, epithelial cells migrate over a wound to heal it, and in axolotls this is also the case. And actually there's expression of a particular gene called amphiregulin, which is observed only during the early early stages of wound healing. And actually, if you overexpress this during regeneration, you end up with a similar phenotype to before. So it seems as though there's an important regulation of whether it's in a wound healing state or in a regenerative state, and there's a difference between the two, and that's what I'm really interested in. So just to recap, axolotl regeneration occurs in stages. After amputation, you see the formation of a wound epidermis, and then blastema cells are specified throughout the limb. They migrate and coalesce to the point of the limb, where they begin to form a blastema. This blastema grows and proliferates, and eventually gives rise to all of the tissues of the limb. I'm particularly interested in this part here because this represents the wound healing to blastema formation transition. And I believe that angiogenesis, or the regulation of angiogenesis, is important in this process. So just a quick run through what angiogenesis is. Angiogenesis is the process of forming new blood vessels from existing vasculature. So here we have a blood vessel here, which in response to pro-angiogenic signals, such as vascular endothelial growth factor, some cells are able to respond to this, change morphology, and begin to migrate towards the source of the signal. And they also pull along other cells with them, and ultimately give rise to a new vessel. And here you can see this. This is from a zebrafish embryo. You can see this happening in real time here as the cells migrate towards the source of VEGF signaling in the dorsal part of the embryo. So this happens by VEGF binding to VEGF receptor in endothelial cells and inducing pro-angiogenic signals. So what I'm doing now is I'm trying to block this process by using chemical inhibition of VEGF signaling. So I applied this to axolotls. Sorry, so the hypothesis here, I showed before, is that angiogenesis is required after wound healing to allow proliferation to take place. So I applied this to axolotls and took sections of the regenerating limbs, and here in this control animal here, where I used HCR in situ to stain for endothelial cells, you can see that there are endothelial cells in red in the blastema. But when we block VEGF signaling, we don't see any really up here. I can take a zoomed in image here, you can see this in more detail. So we were able to block angiogenesis, which is what I was trying to do, and we quantified this by measuring the number of vessels in the blastema. So if you take a look at this in a gross morphological way, you can see that the blastema is actually smaller in AV951 VEGF inhibited animals compared to controls. Actually, if you push this through to 22 days, you'll see that actually there is no regeneration at all. So it's unable to form any of the tissues, and it actually stays as a small bump just here. So it was very interesting, but I wanted to know why this is happening. Does it actually form a blastema? So our lab identified CASL2 as a blastema marker. This is only expressed during regeneration. It's not expressed in the limb normally. And it's very robust, always labels the blastema. And you can see here in the control, the blastema cells are all labeled by CASL2. And then when we look at AV951 treated limbs, we also see that the blastema contains CASL positive cells. So the specification of blastema cells is not affected. It does seem though as though the migration is slightly disrupted, and we see more cells that are not quite making it to the blastema. But it does form, the blastema cells are definitely specified. So I wondered whether it's due to proliferation. So I used EDU staining to label proliferating nuclei. You can see in the control there are plenty of proliferating cells going on. And then when we look at VEGF inhibited limbs, we see that actually around here we don't really see any. And if we quantify this, we see that there's a significant reduction in proliferating tissue in the blastema itself. But if we look actually at the rest of the limb, there's no difference. It seems like it's a specific blastema defect. So I wanted to know what was going on in a bit more detail. So I did some bulk RNA sequencing, and unsurprisingly, I found that angiogenesis was differentially regulated, but also that we saw this difference in epithelial differentiation. And actually, AREG, which I mentioned at the start, was differentially expressed. So I went to validate this by HCR, and we see that in the control, we don't really see much AREG expression at 11 days post-amputation, whereas in the AV951 treater, we see this expression of AREG, which is not normally there. I'll just zoom in here so you can see the list a bit more. So this suggests that perhaps the wound healing response is still active, and that without VEGF signaling or angiogenesis, perhaps it can't be resolved to allow the blastema to progress. And this kind of makes sense because there are a lot of papers that suggest that AREG induces VEGF signaling, so maybe it requires angiogenesis to resolve this. So this suggests there is a temporal requirement for VEGF signaling in the transition between wound healing and regeneration. So to summarize so far, if you inhibit VEGF signaling, we see a loss of regeneration, impaired blastema proliferation, and reduced blastema cell coalescence. And also we see this potentially scar-like wound epidermis, including expression of early wound healing genes like AREG. So it seems as though there's probably a process here that goes from wound healing through to blastema cell specification, and then it requires angiogenesis in order to move into blastema proliferation and growth. So I'm going to talk a little bit about future aims now. So I didn't realize how long it took to make an axolotl transgenic or mutant when I first started this project, but it takes about 3 years. So axolotls are pretty slow living animals. So from injection of an embryo to try and make a transgenic, it takes 1.5 years for them to reach mating age, and then another 1.5 years to get the stable line. So I'm in the process of doing this right now. So that took a back burner while I tested out the chemical inhibition. But it seems as though we're starting to see endothelial-specific transgenics, which hopefully we can raise and will transmit to the next generation. So importantly, these transgenics also contain a specific receptor to allow viral integration into those cells only, which will enable us to do to not only use the GFP to identify rare endothelial populations potentially using enrichment with fact sorting and single-cell sequencing, but also using the virus to perform lineage tracing and see whether endothelial cells are capable of giving rise to other cell types. I can also potentially use this to deliver DNA to test endothelial blastema interactions and see how endothelial cells interact with other blastema cells and regulate this formation, this growth, and whether the same with wound epidermis as well, and see how they interact with each other. So I'm running a bit fast, sorry. So with that, I'd like to thank the HFSP for funding the project, and my lab for supporting me, and everyone that's been a part of this project, including Ryan, Kelly, Julia, Hani, Alexandra, Stephen, and Adnan. So I'm a little bit running ahead, sorry, but thank you.
Thank you so much. We have time for several questions.
Hello. Hi. Very nice talk.
Thank you.
Is VEGF receptor expressed only on endothelial cells or also on other cells?
So that's a great question. Actually, that's something I'm looking into that. Yeah, so we're actually looking into that at the moment, and it seems as though some VEGF receptors might be expressed on blastema cells, and actually we do have some evidence that I didn't talk about today because it's very preliminary, but it seems as though maybe blastema cells are responding to VEGF.
Yeah, because also you see it on the apical side, the fact that you have EDU reductions, it seems like actually that it's only those cells that are responding.
Exactly, yeah. This is what I'm thinking, yeah. So it seems that maybe there's a differential regulation of EMT genes as well, so yeah. Thank you.
Here, very nice talk. I just thought whether you have considered to make spatial transcriptomics of regenerating limbs. In this case, you would identify, apart from endothelial cells, probably other cell types and specific factors that would facilitate regeneration.
So yes, the lab has tried to do this in the past. We've hit a few blocks there. The axolotl genome and transcriptome exist. They're not great. There's still a lot of work to do on those, and actually a lot of the collaborators we've worked with to try and get spatial working have been kind of put off by the transcriptome and the mess that is the axolotl genome. So we keep pushing these collaborations, and we are trying to move this, but it's something that I've been wanting to do for the last 2 years, basically. Thank you.
Any other questions?
Yeah, fabulous talk. I had a question. So do you know how does the wounding response drive the angiogenic capacity? And my second question is that if angiogenesis was a key driver for regeneration, how did we lose it in other tissues that have angiogenic potential but don't regenerate?
Okay, great questions. So to answer the first question, sorry, could you repeat the first question? Sorry, I'm just drawing a blank.
What is it in the blastema that drives angiogenesis or the angiogenic potential?
That's right. Okay, so actually we think AREG might be involved with this, and actually like EGFR signaling generally. So it seems as though AREG is upstream of angiogenesis in a lot of tissues, and actually when you overexpress AREG, maybe you get too much angiogenesis. There's evidence from the mouse that if you have too much angiogenesis during digit tip regeneration, it blocks regeneration. So it seems like there may be this temporal control of this. And then as to why we lose the regeneration capacity in vascularized tissue, that's a great question. I think there's a bit of a trade-off probably between both like patching up a wound quickly and regenerating, depending on the organism. I think the regenerative response is kind of still there in mammals, but it's kind of squashed by this rapid wound healing response. So yeah, maybe if there's a lot of angiogenesis, that kind of just does that. Yes. Thank you.
Okay, thank you so much for a fantastic talk. We'll move on to our next speaker.
Thank you.